I would like to do later in my career, and also give you just kind of some background of how I've gotten here. So my background is through sort of a traditional research path. I went to undergrad thinking I was going to go to medical school. I had a really like transformative research experience in a virology lab, which I don't like virology at all, and I didn't want to do that, but that was my opportunity, and I just loved all of the process of doing research. And I had already like taken MCATs, but I was like, I think this is actually the thing that I wanted to do. So I switched paths to apply to graduate schools to PhD programs, and did a PhD program in neuroscience. And in that program, I studied cognitive decline, and that occurs with age, and did a lot of research on non-invasive neuroimaging techniques that were looking at cerebrovascular function and vascular health in the brain. And we were seeing a lot of observational effects that kind of pointed to the idea that the vasculature was really involved in the cognitive decline that occurs later on. And so I really became convinced that the best way to approach some of these diseases of aging or cognitive decline was through prevention. And that sort of brought me into this space of natural medicine, which focuses a lot more on prevention in healthcare. So yeah, now I'm a postdoctoral researcher here. I'm in an R90 program, which is funded by the National Center for Complementary and Integrative Health. And the program I'm in is really cool. It has the goal of training translational researchers in complementary and integrative health approaches, like me, because I haven't been exposed to a lot of these approaches. And the mentors affiliated with this program actually span across multiple institutions, including the Oregon Health and Science University that's up higher on the hill, and botanical research going on there. They have a center there, which I'll talk about in a second. And then this talk that I'm going to give that I've planned for today, it's going to be a lot more about the research design and sort of the process of doing research. I'm focusing more on the parameters of doing the research, but I'll leave my email up at the end. And if you're interested in chatting more, or seeing some of more of the data, I'll be showing a little bit, but I'm not focusing really on a lot of the data here. You can contact me afterwards and we can continue discussing. And I'd really love for this to be informal, structured in a way that whatever like serves you best. So like feel free to interrupt and ask questions, even if those questions are not really having to do with like the research that I'm showing on the slides. That's fine. I'm happy to talk about like path, or if you have questions about what it's like to do research, that's fine with me. So I'll get started here. So all of the research that I'll be talking about today is a small part of research funded through this BENFRA Botanical Dietary Supplements Research Center. This is located at OHSU. And the BENFRA is one of 3 major botanical dietary supplement research centers funded by the NCCIH and the Office of Dietary Supplements. So they've like combined forces to fund these centers. BENFRA stands for Botanicals Enhancing Neurological and Functional Resilience in Aging. And the goal of that center is to investigate promising botanicals that are already being sold. They're already on the market, sold as dietary supplements, and they're reputed to improve resilience to age-related changes in cognition, sleep, or mood. And the idea is that ultimately the work will hopefully support the development of more optimized products and with more reliable health benefits by kind of understanding the important biological and chemical properties of those botanicals and building an evidence base for their use since they're already being used. The center is mainly researching 2 botanical herbs. And that's quite amazing to me because there's a lot of researchers on this, but it's just like focusing on these 2 things, ashwagandha, and which I'm not going to be talking about at all in this talk, and Centella asiatica or Gota Kola, which is the focus of this talk. Yeah, go ahead. I don't actually know them very well. Yeah, I should learn what their focuses is. But you can go on. They're called the carbon centers, and they have the grants that they support there. So my background and interest is in the neuroscience of aging, which seems like particularly important. The aging population continues to grow in both the United States and the world. And we know that dementias, which are considered pathological, like Alzheimer's disease, are much more prevalent in older age. That's like the main predictor of these things. And even in normal aging, like outside of pathology, without any like biomarker signs of it, there are often declines in cognitive ability across a number of cognitive domains. So that's what I'm showing here. This figure is showing the typical cross-sectional pattern that's observed. This T-score thing is just a way to standardize scores across different items. And I just want to clarify, because some of you will have taken stats now. And this is kind of confusing because the T-score is like a common statistic that you learn about in stats, but this actually means something different in psychometrics. So the psychometric T-score is just a specialized term where the average score is 50 and the standard deviation is 10. So it's just a way of sort of normalizing. And what you usually see is the separation between domains that are considered part of what's called crystallized intelligence, which is supposed to reflect the accumulation of knowledge and skills that you've learned and practiced throughout your life, and those that are considered fluid intelligence, which reflect the ability to reason and sort of think flexibly, often about new problems. So fluid intelligence tends to decline more with age, whereas this crystallized intelligence seems to stay stable or even increase, such as for these like verbal ability or numerical ability. And I always show this because when people give talks about cognitive aging, they almost always just show this cross-sectional figure, which is strikingly depressing, I think, which is really good for getting funding, getting people scared. But if you look at longitudinal trajectories, you usually get a more optimistic picture. Perceptual speed still seems to decline pretty linearly across the lifespan. But for many other domains, there seems to be good stability on average until around the mid-60s of age. Kind of either way, we observe that in later life, cognitive ability tends to decline, although there's a lot of variance in this. And like where there's a lot of variance, there's a lot of opportunity to increase our understanding of that particular thing, like why is that happening in some cases and not others. Yeah, good. So this is looking at cross-sectional responses. So and this is looking at longitudinal responses. So cross-sectional means that we're looking at a bunch of different people that are a bunch of different ages, and longitudinal means that we're following the same individuals over time. So yeah, I was going to stop here and ask, just to like put on our thinking caps, like this is a part of doing research, is trying to understand what are the biases that we bring to the table when we look at data in different ways. So do you have ideas of why these two pictures look so different, even though they're investigating the same outcome measures, they're the same cognitive abilities? Oh, what's wrong? Any ideas? We have one case where they're following people over time, and one case where they're just taking a snapshot of a bunch of different groups of people. So you're suggesting sort of what the pattern is showing, I think, that when you look at individual people, you're seeing you're seeing a different pattern than if you look at a bunch of different people. But even if these samples are the same size, including the same amount of variance among people at the baseline, their scores still look different, like these trajectories still look different.
for your question, but it's really interesting to see all these things moving. I mean, most things moving till age.
Yeah, exactly. I think that I like showing this because even like you can see these are from the same paper, A and B, and I feel like everyone just shows A in talks and like ignores B. And I think it's important because this is optimistic and kind of like in some ways you think the longitudinal data like following a person over time might give you maybe a more accurate picture of what's going on. But both of these methods have biases incorporated in them. So with the cross-sectional shot where we're subject to cohort effects. So different generations, groups of people go through different experiences in their life that change like large aspects of their abilities and how they think about the world and a lot of different things. So the idea here is that people that are currently 81 might have had a lot of different factors in their life that would cause them to have lower performance than if someone born now like ends up being 81. They might have had less schooling. There might have been less focus on certain aspects of schooling that like promote flexible thinking. They might have played less video games. Like there's a lots of different things that could factor in. And then also though, for the longitudinal studies, you're also subject to some biases. So we know that there are strong practice effects. So if you're taking tests, even if they're different tests, but they're sort of in the same domain over time, you improve on those scores. So that could be part of why we see some improvement here. We also are subject to perhaps losing subjects that enter the study but start getting sick or performing worse. They'll stop doing the longitudinal data collection. So there are things that we need to think about in how we collect data. What's considered normal age-related cognitive decline sometimes transitions into mild cognitive impairment or dementia. And the cause of dementia is probably multifactorial with certain factors beginning quite early in the lifespan. So this visualization concept came from a researcher named Clifford Jack originally. And I really like it as inspiration to work on my graphics because the original paper, like pretty much just based on displaying a graphic like this, now has like almost 5,000 citations. And the idea is that in Alzheimer's disease, there are a number of different like measurable parts or measurable markers of the pathology. But if you watch people over time, it looks like some of those measurable markers will occur before other ones. So for instance, you see this like amyloid plaque buildup. It typically precedes the neurodegeneration and like cognitive symptoms that really define the disease. It happens a lot earlier in time. The body, though, does a lot of maintenance on its own. This is a concept called salutogenesis. And one idea that I like is that it's easier to promote like maintenance processes before the mechanisms of pathology really start to take hold and make that harder to do because they're interacting and interfering with some of those salutogenic processes. So my opinion is that we really ought to target much earlier for an intervention. That's where my sort of preventative outlook comes from. And it's hard to do that for a couple of reasons. One is that many of the drug approaches that target the pathologies that we know about carry some risk. So the benefit to risk is worse when you don't know what percentage of your group is going to go on to develop the pathology. Like we don't want to risk people's lives too much if we don't know that they're ever going like on the trajectory to get sick. And unfortunately, the further you go out this way, the murkier the causation gets. Like not everyone with amyloid plaques goes on to develop dementia. And a second major one is that doing a trial here, like a clinical trial to prevent or prolong dementia would be extraordinarily expensive. So that's why even though a lot of physicians, both in natural medicine and in allopathic medicine, believe that prevention is like a good idea, there's not a lot of clinical evidence showing these longitudinal prevention studies. But I think a really good trade-off is to target earlier risk factors of Alzheimer's and other age-related dementias. And cerebrovascular impairment actually occurs quite early and seems like a good candidate to me. You see some cerebrovascular impairment in nearly all like postmortem Alzheimer's disease brains, and it's a major risk factor for future dementia. And then we have techniques to measure it and intervene on it already quite safely. So that's where my interest lies. The last graphic was somewhat, this is like theoretical, like I just drew this, but it's really hard to do long enough longitudinal studies to look at these things, but there's some hints that from like observational data that this viewpoint is true, that support this view. So this is from a paper from a really large data set from the Alzheimer's Disease Neuroimaging Initiative, and they quantified the level of biomarker abnormality for different stages of disease. So this is like early mild cognitive impairment, late mild cognitive impairment, late onset Alzheimer's disease, and HC just stands for healthy control. And they map these trajectories of the biomarker abnormalities through these stages, and finalizing with the diagnosis of late onset Alzheimer's disease. And what they see is some significant separation of these biomarker trajectories, even like quite early in the early mild cognitive impairment category compared to outright Alzheimer's disease. And what they see is that like their marker of cerebrovascular function, cerebral blood flow, this biomarker was the earliest predictor of all of the predictors that they looked at for Alzheimer's disease. So I think there is some evidence that this is something useful to focus on. So I'm interested in finding like safe, potentially long-term intervention strategies that modify cerebral vascular function for those reasons. And botanicals, I think, are a good candidate for this, that may promote resilience to decline in neurological function. And one of those ways is through modulating cerebrovascular health. So when you study botanicals in humans, there are actually different regulatory pathways depending on your goals. You can study dietary supplements, and that's how I'm categorizing what I'm doing. If you're interested in like maintaining health by modifying physiology associated with physical and psychological stressors. But you can also study a botanical as a botanical drug if your goal is to reverse or slow a disease process. And I find this like they're quite different regulatory pathways. You might need an investigational new drug application with the FDA. You have to liaise with them a lot. There's a lot more protections if you're developing a botanical into a drug. And I think it's really interesting to think about these pathways in the context of age-related diseases in particular, like I'm doing, because they have especially age-related diseases that have risk that are modified a lot by lifestyle factors, because it's kind of unclear. Like I'm focusing on a dietary supplement that probably improves physical and psychological responses to stress, but long time in the future, that probably will be preventative to some kind of disease state. In either of these cases, though, there's really a strong need for high-quality clinical trials to support and the use of these botanicals or their development into pharmaceuticals. Yeah. So I'm bringing this up because this Benfra work that I'll be showing you is meant to fit into this framework working towards developing these high-quality clinical trials. That's like the end goal of this work. And this is a framework specifically proposed by the NCCIH. And the roadmap basically emphasizes that a significant amount of good work needs to be done before you jump into designing efficacy trials. It's really hard to have a well-designed, well-justified efficacy trial. And they had a lot of early on failures in replicating or finding good results with efficacy trials. So they've started breaking it down into these blocks. So you start by looking at historical use, gathering fundamental mechanistic preclinical knowledge, and then you can sort of work up through these other building blocks to the eventual goal of the efficacy trial. And you can work on some of these blocks in parallel, but the levels really build on each other. So it doesn't make sense to work on recruitment and retention before you figured out and sort of justified what population you're even going to be looking at. or what outcomes you're actually even going to measure, because then you would have to go back down if those turn out to be different. And the Botanical Research Center I'm part of is doing a lot of these building blocks in tandem, but basically with the goal of taking a botanical through all of these steps of product and dose optimization, understanding the mechanisms, evaluating the botanical in vivo, so like in animal studies, and then establishing translationally relevant biomarkers, and then finally into the design elements of clinical trials. So this is part of the reason why they're only studying just a couple of botanicals. It's like a tremendous amount of work that you need to get here. And my place in this pipeline is really in this space of evaluating in vivo efficacy on some cerebrovascular function measures, because that's my like personal interest, and then translating them into human trials at those early stages of age-related cognitive decline. So like I said, I'm doing work on Centella asiatica, which was traditionally used for brain health in Ayurveda. It's now widely sold as a supplement, most typically marketed as Gota Kola. And you can get it in all sorts of forms: powders, leaves, liquid extracts, even in creams. It's actually most commonly used for as a skincare product, because the highest amount of evidence is that it's useful for like wound healing and some other aspects of skin health. And there are a bunch of branches within this Benfra Center that are collaborating together to like take the botanical, they grow and assure the quality of the Centella extract. They study the compounds in the plant material, in plasma. They conduct safety studies in both animals and in humans now. They have these in vitro studies to try to look at effects in primary neurons. And then in vivo studies in mice and also fruit flies, looking at behavior and potential mechanisms underlying changes to behavior. So there's a lot of different groups, like many groups, working on studying the same botanical. I haven't done any of the behavioral research myself, but I think it's important to show you some of the background of benefits to cognition in mice, because those studies showing changes in cognition are like the main justifications for taking the next step of investigating the physiological mechanisms that might underlie those benefits. So this is a study looking at 20-month-old mice who receive either regular water, these control mice, or water that's treated with Centella inside it. And they're treated for 5 weeks, but they undergo a battery of cognitive testing that begins in the second week of their treatment. And this is an example result from that study on a task called the object location memory task. So in this task, mice are habituated to an environment with 2 objects. And then during a test phase, one of those objects is moved to a new location. And a mouse with intact memory will explore the object with the new location a lot more than the object at the old location. And you do the first test after a 2-hour delay, but you can make this test even harder by incorporating longer delays, like this is a day. So they tested a 3rd location at a 24-hour delay. And then these are the results from the 2-hour retention and the 24-hour retention. And what we see is that in the 2-hour retention, the control animals explore the novel location more than the old location. So these are control mice. I guess this is particularly for the control females. It does look like the females do this better than the males at baseline. But giving these old mice Centella improved the performance for both the male and female mice. And then after the 24-hour delay, actually neither the old females or the male control mice showed any preference for the novel location, which we interpret as like they have bad memory. But then if you treated them with Centella, they showed a pretty strong preference for the novel location. And then more recently, the Benfra Center replicated this result in a similar paradigm. This slide is showing one example of that from a test called the novel object recognition task. So this is really similar. Mice are placed in an environment with two objects, just like before, they become familiar with. But this time when they're tested, one of those objects is replaced with a novel object instead of a novel location. I'm not sure why they chose to change the test here. I haven't asked. But they added the addition of a young control animal along with the older control animals. And the centella was also given in two ways, either in the diet or in their water. And what you can see in this test is that without any sort of treatment, young mice do pretty well. They spend more time with the novel object. The older mice seem a little more impaired. But if you give the old mice the centella extract in their diet for 5 weeks, looks like they might have a little improvement, but not a significant amount. But if you give them the centella in their water, they have a significant improvement and they look pretty similar to the young mice. So we learned from this that the route of administration seems to affect the bioavailability of the centella and the effect of the treatment. And we followed up with that and found that that's true. It's more bioavailable in the water. So what is it about centella that's so helpful? We have two main leads. There's this class of molecules called triterpenes that are fairly unique to centella and have some beneficial properties that have mostly been studied in relation to the skincare that I was telling you before, such as like collagen synthesis and free radical scavenging. But pure formulations of triterpenes also seem to have some vascular benefits, such as in like venous insufficiency. And then more recently, there's some evidence that these compounds called caffeoquinic acids may activate NRF2, which is a transcription factor that regulates the expression of a bunch of genes involved in the antioxidant response. Anytime I give a talk about centella, like the main question I get is if I like take the stuff myself. So I'm just gonna head that off here. And I think it's like an interesting point because not all of the centella asiatica on the market that's sold have these compounds. We've been testing a lot of different ones because I'm interested in like using some of those in different studies. And most of the products don't publish their triterpene or caffeoquinic acid content. So it's kind of hard to tell what you're getting. Some companies at least claim to standardize their extracts to certain triterpene level content. But if you're interested in trying it, I would just say that you have to be careful because like not all the products are equal in quality. And because of that, I'm also kind of like holding back until we know more about like what a good product is. So my tiny part in this giant like machine is working on some of the in vivo experiments on aged mice. And we're interested in exploring some of the possible mechanisms that may underlie these cognitive effects. These mechanisms probably interact with each other, but we've chosen experimental measures that are particularly sensitive to mechanisms of cerebral vascular function and brain perfusion, oxidative stress, and neuronal mitochondrial function. And all of this is ongoing. So if I get to data, I'll be presenting data just collected from 20 mice out of a planned 96 total mice. So it's very early on, very preliminary sample of the whole data set. And that's why I wanted to focus more on like what we're planning to do. The sample size is fairly large because we're collecting data from both treated and untreated mice, 3-month-old and 18-month-old mice. So like that's considered like a young adult and maybe around like 65 or so, older adult. And male and female mice. And we're interested in all of those factors. So we need to do a lot of mice in order to be powered to do those analyses. And I think one of the really, to me, powerful aspects of this study that you don't see that much in the research literature is that we're going to be able to correlate all of these like different measures like across all of the measures because we're getting each one on all of the mice. So that it's pretty exciting to me to have this sample. So the mice arrive, we treat them for 5 weeks, just like before. We're using the exact same protocol with the treatment in the water with the same product to try to match those behavioral results. And then after 5 weeks of treatment, I do magnetic resonance imaging on the mice, where I measure blood flow, anatomy, and proton spectroscopy. The mice recover for 2 days, and I do a series of additional experiments. I use a technique called laser Doppler flowmetry to measure neurovascular coupling. So that's like how much the vessels react to neural activity. It's important that they do that to bring in blood flow and nutrients to the active tissue. And I do that with by measuring from the contralateral cortex to a hindpaw stimulation. So I'm like sending in little electric shocks while the animal is anesthetized. Then I euthanize the animal, collect plasma for an oxidative stress assay, and then I simultaneously extract the brain and do live ex vivo imaging to see how vessels in those brain slices will react to an oxidative stress challenge. So I can keep the brain tissue alive for about 5 or 6 hours and do these experiments on the vessels. And I always like to show like faces of the teams that I work with. So this is the main team that I work with day to day. And this is Amala. She's like the program director for the whole Benfra Center and the administrative core lead. And Nora Gray is the lead on all of the in vivo work. So like my particular slice of the experiment. This guy, Marty Pike, trained me how to do mouse MRI. My background's in human MRI, and it's quite different, actually, to like the things that like mice don't do what I tell them to do in the same way that the humans do. He's also given me really good mentoring on how to be successful at crabbing on the Oregon coast. So I really value that relationship. Manoj is leading the analysis of the spectroscopy. And then Anusha is leading those ex vivo experiments with the brain slicing. And Laura is a research assistant in her lab who is just great. One of the graduate students referred to her as the beating heart of the lab recently. So she's been really instrumental in helping me manage the treatment of the mice and helping me collect the mouse plasma. It's quite a lot of work. I didn't like mention it, but I get through 2 mice every week with like a whole week's worth of work. So like the magnetic resonance imaging is a whole day, and then these other experiments are a whole day per mouse, and then I need about a day of analysis. So to get to the 96 takes like a tremendous amount of time and work. So I'm going to talk about the MRI measures. If you've ever seen an MRI machine, you know that they're like pretty big. And I think that people are always surprised to learn that our mouse MRI is like even bigger than that. The bore, which is like the tunnel where you put the animal, is much smaller. But since the mouse brain is so much smaller than a human brain, you actually need like a much more powerful magnet to get the same spatial resolution of that brain. So it's like this giant piece of equipment. We collect a T2 weighted anatomical image. We collect proton spectroscopy from 2 areas, the striatum and the hippocampus. And we collect 2 measures of blood flow in the brain. One of these measures gives us the mouse's resting blood flow from one slice of tissue. This is an example of what those like perfusion maps look like, and we're just collecting one slice. And then I also received a pilot grant through the neurology department at OHSU to add on a measure of that's called cerebrovascular reactivity. So in this measure, a mouse is exposed to carbon dioxide enriched air. And in response to that hypercapnia, all of the vessels in the brain dilate near maximally. So it gives us some sense of the vasodilatory capacity of the vessels in the brain. And I call that relative change from the baseline blood flow to whatever that maximum is, the cerebrovascular reactivity. I've been mainly working on that blood flow data, but my colleague Manoj Sami is an expert in the spectroscopy. He's begun analyzing this proton spectroscopy data. And by analyzing this MR spectral data from different brain regions, we get insights into the metabolic status of the sampled region. So the y-axis is the intensity of the signal at different chemical shifts. And really all you have to know is that that's like related to the concentration of a particular metabolite. So if these peaks are bigger, there's more of that metabolite in the tissue. We're mainly interested in the ratio of NAA or N-acetyl aspartate to creatine. And this has just been shown to be like a measure related to neuronal health and mitochondrial function in the neurons. So we're looking at that. I've been looking at blood flow, and I want to emphasize that these are very preliminary results. When we look at resting cerebral perfusion, that's what I'm showing here. We see a pattern where older mice have slightly lower perfusion than younger mice, and mice treated with Centella have slightly higher resting perfusion than the untreated controls. Since this is like really preliminary, and since we have such small numbers, I'm collapsing the other groups in each of the figures. So like all of these animals are the same in these two plots. So you can see the other groups represented as individual points in each graph, and the color code is down here. So for instance, like the mice treated with Centella here are represented in yellow, and the controls are represented in purple. But I'm showing the mean group effects. And in the statistics where I'm looking at these effects, I'm controlling for the effect of age and these treatment effects, and I'm controlling for the effect of treatment in the age effects, and neither of them are coming out significant right now. These are similar figures, but this time looking at that % change in cerebral blood flow in response to hypercapnia, so the cerebrovascular reactivity. And we can see again that the older mice, they seem to have slightly lower cerebrovascular reactivity than the younger mice, maybe, although there's not that much of a difference here. But with the Centella treated mice, we're already seeing that they have slightly higher cerebrovascular reactivity than the untreated mice. And this effect is already coming out as statistically significant. So this is very exciting to me. I'm trying not to make too much out of it, since like we still have a lot of the sample to collect, but it seems like it might be a really strong effect. And the most recent analysis that I'm doing is looking at volumetric analysis on some of the particular regions of the anatomy. So in this plot, I'm showing the legends down here, and applies to all of the plots. I'm showing young and old, control and treated animals as 4 different groups. So like the 3-month control animals are green, 3-month treated are blue, so that you can see all of them. So there's only like 3 or 4 mice in each of these groups, not to make too much of it. But I'm excited because it looks like, depending on the region I'm looking at, there's sort of different like patterns of effects, and some of them match what has been shown in the literature. So like for the whole brain volume, I'm seeing not really that much of a Centella effect, but I am seeing an age effect, and that replicates what other people have seen. Whole brain volume tends to decrease in mice with age. In the hippocampus, you typically don't see that much of an age effect. In mice, you do a lot in humans, so different animal, different results. But I am seeing a Centella treatment benefit in both the young and the old mice in the hippocampus, and I thought that was cool. And then some other regions like the cortex, I seem to see a decrease with age, which has been seen before, but also perhaps a little bit of benefit of the Centella effect. So I think these, like, the conclusions that I'm currently telling you are like way too specific for comparing, 3 to 6 mice in each group. But I'm excited about the potentially different patterns of interactions between age and treatment that might emerge from the different regions in the brain. And then, so after the imaging, again, we do these other tasks, the second part of experiments. In these, I measure neurovascular coupling by looking at the changes in flux of blood in the brain after a hindpaw stimulation. And that's kind of like an example of what that change in blood flow looks like to me. And then I euthanize the animals. Typically, Laura, the research assistant, will help me by collecting the plasma to look for oxidative stress measures in the plasma. And then I simultaneously prepare these brain slices to investigate how the vessels react to an oxidative stress challenge. So in this challenge, I just apply hydrogen peroxide at very low concentrations, about 1 millimolar. Well, I guess some people would consider that a high concentration, but I apply 1 millimolar hydrogen peroxide to brain slices. The capillaries tend to constrict in response to the oxidative stress. So our hypothesis was that there would be less constriction in the centella-treated mice compared to the control mice because they have more resilience to the oxidative stress. This is a short video of an example vessel just to show you like what the constriction looks like when I apply hydrogen peroxide. I'll just orient you first because like to me, I stare at these all day. So I'm like, oh, this is obvious what we're looking at, but I've learned that it's not. You can hopefully make out a capillary lumen here. So this is one of the walls. This is another one of the walls. It kind of bifurcates here and then the wall's going up here. Are you seeing that kind of? Okay. Good, I'm not imagining it. And I'm gonna play a video. It's sped up so that 17.5 minutes of recording is compressed into 7 seconds. And I want you to pay particular attention to this area where there's a type of cell, contractile cell called a pericyte is located there, which will constrict that part of the capillary after I apply the hydrogen peroxide. So it's playing now. I apply the hydrogen peroxide and you see that constriction. And one thing that's interesting about this I'm sort of exploring more that was totally unanticipated. I love stuff like this in science. Like that's to me, the cool it's nice to see that like your hypothesis is maybe supported by your evidence and you can write papers about that and get grants, but it's always fun to like find something that you weren't expecting at all. And we're definitely finding that here. I'll play it again. You'll see when we apply the hydrogen peroxide, it constricts, but then even when we continue to apply the hydrogen peroxide, the capillaries will eventually sort of like relax. That seems to be an effect that happens more with the older mice, happens more with the non-treated mice. So we're sort of interpreting that as like an inability to hold on to the constriction, and no one knows why that might be. Like there's no we're like, oh, we should study why that happens. That's like a new interesting measure. So these are the planned next steps. I'm really excited to continue the work and collect the fully powered sample of 96 mice. And in the near future, I'll be analyzing these laser Doppler flowmetry measures, the oxidative stress measures. I'm excited to start correlating the different kinds of measures together to see if differences in things like the oxidative stress maps onto those differences that we're seeing with the vascular function. And also investigating further that mechanism of the release of constriction that we're seeing in the hydrogen peroxide experiments. And I'm starting, so I have a pilot trial ongoing investigating the centella supplementation on human peripheral endothelial function here. And we'll also be writing a early career mentored grant to continue this work in humans, looking at translating these imaging measures to the human measures. So this is just acknowledging like doing research like this, even in my little piece, takes a tremendous amount of support and help. So this is just acknowledging all of those groups of people and different funding sources, including the National Institutes of Aging. Even though this isn't an NIA grant, they're generously allowing us to use their aged rodent colony, which is amazing. Yeah, I just like asked them for mice and they ship them to me for free, which is really cool. Yeah, so I'll take any questions. We can have a discussion about this. Also, I'm definitely interested in finding students who might want to like do research, be involved. I'm very open to doing that in any capacity that like works. I would love to get more students involved in some of this stuff, or if you have particular interests, that I'm open to that too. Here's my email and some links to other things that I've talked about if you're interested in knowing more. So good. That's all. Questions?
If you have any questions, I can give you the mic.
Looks like.
Okay, well, thank you for coming. My name's
Yep.
Danielle Zuzero. I'm a student here and a part of the research program. And this is Research Week. Thank you for coming. We have another talk tomorrow and Thursday, and then there's student presentations all day on Friday. There's posters outside. And yeah, we just are hoping to foster a community within the research world. So thank you, Ben, so much for kind of launching that for us. Standard Process is a sponsor of the event, so thank you very much. Yeah, and the Student Government Association also. So thanks for being here. Yay.
Can you talk a little bit about some of the human pilot trials that you're going to be doing and what you're going to actually specifically what you're measuring?
Yeah, so the trial that I'm starting already is looking at a peripheral endothelial function measure. So the idea is that a lot of these things that I think are happening in the brain are stepwise. Like if we're seeing dysfunction in brain vascular health, typically that in itself is preceded by other vascular factors in the body. So you'll have like stiffening in some of the arteries that starts very early on in the lifespan, some stiffening, and that leads to peripheral endothelial dysfunction that's sort of continuous. So we can measure that. We have these little like balloons that we can put on a finger, and then we can restrict blood flow to through the brachial artery. And when we release that restriction, there's a response, a normal physiological response to that, where blood flow is massively increased to support the needs of the tissue there. Now that like, oh, the blood flow is released, and we can look at that as like a measure of the endothelial function capacity. So I'm looking at that. That's just a small study with 30 participants, and kind of getting my feet wet and doing my own clinical trial research. Yeah. And then I'm for my mentored K award, I'm planning doing imaging measures directly in humans.
How will taking Centella?
I'm having them take it in capsules for ease. And that was after some discussion, because in the mice, at least, we're seeing that it works better in the water. So we're just increasing the doses a little bit, hoping to get the same availability. Yeah.
Any other questions? Great. Well, that concludes our first research team presentation of Research Week. If you attended and want to enter for the raffle, there's raffle tickets back there, and you'll find out if you won on Friday. And yeah, thank you, Dr. Zimmerman.
Thank you.